helpful. Obviously, it will be the costly and need more and more time, but I hope that in future this techniques it will be the helpful. Just like you can say Vadyachar or you can say Yogachar or some of the Ayurvedachar, they are developed the medicine on the basis of the personalized basis. This is known as the you can say dispensing, and the now dispensing is the most probably the absolute, and we are the working on the pharma technology. But even that some of the disease require to a personalized medicine and personalized therapy. So I hope it will be the helpful to the all audience, student, faculty member, and researcher. So I think expert is the first time is the interacting with the ISF College of Pharmacy. So I just give only the 2 and 3 sentence brief about this ISF College of Pharmacy. This is the an autonomous college, NAC accredited A grade, and we have the NRIF ranking is the 29 in pharmacy category in India, and we have the excellent research team. And at present, we have the 39 research labs with the well equipped in the digital model of the machinery. So our faculty members and research team is the doing very hard work, and this is the one of the premier institute in the India. So whenever you have visited the India, you're welcome at this place, and I hope that you can also accept our invitation, or you can also come with your team so that we can some do some of the collaborative work also. Our pharmacology department is very strong, and we are running animal house with the breeding and trading facilities and providing the animal near about 10 universities of this area. And we have one of the analytical lab. This is the NABL accredited. So 80 of the pharmaceutical companies samples they are regular coming for the testing like of the purpose. And recently we have developed the zebrafish lab. So I hope that either it may be pharmacological work, synthetic work, and some of the related work of the polymers. So this institute and novel drug delivery system, nanotechnology, our faculty member is the quite comfortable to work on it. So without taking time, I am again welcome today's experts, and I am thankful to the and also welcome all the participants, those who are the connect right now or those who are the watching our YouTube channels. So thank you very much, Dr. Dilpreet Singh ji, for inviting me and provide this opportunity for with in front of the wonderful speaker, very young and dynamic, and I hope that his talk will be the very useful to each and everybody. Thank you very much. Over to Dr. Dilpreet ji.
Thank you so much for your words, sir, and a brief introduction about the college. And without taking much time, sir, I will give you a brief introduction about everyone. So basically, Dr. Irham is completed his PhD from Taipei Medical University, Taiwan, and professional diploma in clinical research from Indonesia. He is a visiting scholar in the basically the pharmaceutical ingredient and traditional medicine National Research and Innovation Center, BRIN, Indonesia. Sir has published around 50 to 55 publications in the International Journal of Repute with an overall H index of more than 15. His expertise in basically the genetic biology, chemical biology, and in the molecular biochemistry systems. So without taking much time, sir, can we start the presentation?
Yes, Dr. Dilfrat is fine. Yes. So can I start now?
Yes, sir.
Okay, thank you so much. First of all, that I would like to thanks to Professor Gupta for a very good welcome speech, and we are honored, Professor, for your invitations, and also our deans and also all of our staff will welcome our collaborations. I think hopefully that next week we can have some meetings, right, and to discuss about the possibility of collaborations. And secondly, that I would like also to thanks to Dr. Wilfred for your kindness and also your patience to discuss uh, about the event today, right? And we discussed a lot uh, about the topic and also about the content of our presentations for today's events. So let me share my PowerPoints or my presentations first. Okay. Okay. Now in Indonesia is the time is 4:30. This means that 1 and 30 hours ahead between Indonesia and India. And today's my presentations is about the precision medicines and challenges in research and clinical implementations. This is my short profile that Dr. Dilprat already mentioned it, that I'm now is the visiting researcher also in the Greens, one of our research center in Indonesia, and also I'm staff in the Faculty of Pharmacy, Universitas of Dahlan. It was located in Yogyakarta, Indonesia. And my field right now is about the pharmacogenomic precision medicines, as well as the bioinformatics and also the drug proposing. So this is my selected publications within 2 years. It has been published in the international journals. Before I'm going to talk in more detail about the concept of the precision medicine and also the pharmacogenomics, I would show you the picture. This picture actually is created in around to 2003 or 2004. It's a long period of times. So the physicians and also healthcare at the time that they have kind of the dreams when the patients come to the pharmacy, come to the apotek. If in Indonesia, is the name is the apotek. They will bring the prescriptions contain the coding of the genetic code of his genetic, as well as that when the doctor came to the ward and then to evaluating the clinical of the patients from the laboratory, they will see the genetic code of the patient. So this is the dreams, but whether this kind of the dreams has come true or not, we can see right now how is that the clinical implementation of the pharmacogenomic as well as the precision medicine is still it's still many barriers. So all the audience, respected audience, we before we are going about the what is the precision medicine as well as that the barriers in the clinical implementations as well as that the opportunity in the research area and also in the clinical implementation, I would elaborate more detail about the first is the basic of genomic and also especially in the human genomes. And secondly, I will present about the clinical pharmacogenomic implementation as well as that the opportunity and also the challenges. First, that when we are talking about the genomic, actually it's not very long period of time. It's around 1987, when it's come first from the editorials with new discipline, new names, and new journals in the Journal of Genomics, in the sentence for the new developing disciplines of genome mapping sequencing, we have adopted the terms of genomics. So the terms of the genomic come from these journals. And what is the genomics actually is the plural of the genes. So when we are talking about the genes, it's many genes. We call the genomics or we call the genomes. And when we are talking about the genes in the single form, we call the genes. So the actually in our when we are thinking about our body, actually it contains several organs, right? And then in every organs we have a trillion of cells, and inside of our cell there is the nucleus, and inside the nucleus there's the chromosomes, and inside the chromosomes there is the DNA. So the DNA is coding of our life that we will talking more detail about this kind of the very small molecule in our body. And this it come from actually the law of the genetics come from the Gregor Mendel in 1865. Along with that, after the Gregor Mendel is found as the father of genetics, there are two young scientists at that time in 1953, Watson and Crick. They found that the form of DNA is double helix, and along with that, after several years, there is three scientists named their name is Nirenberg, Corona, and Holly in 1966 completed the discovery of the Watson and Crick. They found that the form of the DNA is the coding like the A always pair with T and also the G always pair with C. So in details in the future of our slide, we will talking about this kind of the codings. So what is the inside of this kind of the code is we will talking more details, including in the influence of the response of the drugs as well as that for the candidate of drug target genes. And mostly that the scientists when they are talking about the size of this kind of the code, they will use the base pair, kilobase pair, and also the mega base pair to simplify it, to simple to say that this the long of the long of the coding of the DNA. And in our body, then we have around 3 billion letter of the coding of DNA in the human genome. So it was distributed in 23 chromosomes. In every single of us has contains 3.2 billion. So uh, 3.2 billion. It was completed by the Human Genome Project in 1990 until 23. It was it was spent from 13 years ago. It was spent 30 years for discover this kind of the 3.2 billion letters in our body. So right now, actually, we are in 20 years of biology. We call the post-genomic era. Whilst in this kind of post-genomic era, it's in reality, but there is still many barriers in the clinical implementation of the genomic discovery. So in 23 and 24, there is two journals we call the Nature and Science. I think all of us has known about these two journals. It's very uh, very famous journal, right? And then at that time that they they are elaborate more detail in this journal at the time in 23 and 24 about the human genomes, especially in human genome projects. So again, that when we are talking about the genome, I think is we can illustrate that to simplify it and to understand more details about the genomic in our body, we can think about the books. While at that books, for example, they have contain around 23 chromosomes. We can imagine that chromosomes like chapter, and then contain of every chapter is sentences, and then at sentences that we can imagine like the gene. So in this in every sentence contained the letter is like the base pair or the coding of our DNA. So when we are trying to write it down, 3.2 billion letters of our coding of DNA, we will span around 44,200 books. It is very amazing that Dr. Francis Collins is the director of Human Genome Project in 1993 until 2003. They mentioned it that the 3.2 billion letters of our coding DNA, when write it down in 4,200 book, when we are put it in the peltrical forms, it will the height is around 168 meter. It's mean that equal with the Washington's monuments, the tall of the Washington monuments. It is very interesting. So I think when Dr. Dilfred, hello Dr. Dilfred, when you are trying to uh, when you are trying to write it down 3.2 billion letters pairs of our codings, or 3.2 billion letters of our coding DNA, when you are trying to write it down and you spend on uh, uh, 8 hours a day and 60 hours per minute, you will spend on 50 years for to write 6.4 billion letters of our coding DNA. So that triggers Dr. Francis Collins to write the books with the title The Language of God. While the language of God is the genome, is the book of life, and the genome is the livable of the life, the Dr. Francis Collins said in this book. And when we see this very interesting, I think this very interesting illustrations while in the Nature it has been published in 2011. They mentioned about the genomic achievement since Human Genome Project in 1993 into 2003. They mentioned it that so many discovery in every years in every country in the world that so many advance of the achievements. We can see here is the Gregor Mendel's found about the law of genetics, and after that there is the Watson and Crick's found the double helix form of the DNA, and also the content of the DNA is like the ATGC. It was found by the Holly and Nuremberg. And when we can see right now, I think especially in Asian country, there is a lot of the country right now is buildings the DNA biobank. I think it's so far that I know, our country in Indonesia also trying right now and learned from the China to building the DNA biobank, because our population has many races, as many ethnies, that it is important to elaborate, and it is important to find the genetic codings as reference for the future. And when we are talking about the actually about the achievement of genomic, we can divide it into several era, like the pre-genomic era, post-genomic era, and also the application of genomic era. While in this every phases or every era, there is so many achievements, like the in pre-genomic era from 1990 to 2003. There is the understanding the structure of genome, as well as the understanding the biology of genomes. And when right now actually we are trying to implement it in the clinical area that we call the genomic medicines, or clinical implementation of genomics. So we are actually right now is the application of genomic era, but we are still see how that in every country, I think still have problems. And also here, even though in the research area we have many opportunity to identify the many or understanding the biology of disease, as well as that understanding structure of genomes. And when we are talking about the genomic, actually right now the genomic is the one part. We call that the other one is the omics data, like the transcriptomic, proteomics, metabolomics, and also proteomic. We can integrate it between each of them, and then so the purpose is to disease subtifying and identify the biomarkers. And actually, when we are talking about the pharmacy, I think it's very related with the drug discovery right now. So many scientists right now trying to discover new drugs from the information of the omics data that we call, actually I'm right now also developing the drug repurposing based on or driven by the genomic informations, and also integrated with the bioinformatic based approach. And right now, actually, the genomic medicine that we call the clinical implementation of the genomics, they have the several hot area, like the cancer of genomics, pharmacogenomic. When we are talking about pharmacy, I think it's very close related with the pharmacogenomic, because we are talking about response of the drugs that has been influenced by our variation of the gene, or gene genomic variation, that we call the single nucleotide polymorphisms. And the other one is the rare genetic disease. Of course, that the rare genetic diseases still have the problem, because to identify the cause of the disease that we call the biomarker is very important for the rare genetic disease, because for the diagnosis is needed, it's needed a long period of time, right? So how about the drugs? Of course, it will be very have a lot of the barriers to identify the drugs for the rare genetic disease. So in yours, the hot genomic area, or health genomic medicine is very important for rare genetic disease. And the fourth one is prenatal genomic testings, and the last one that it is a very hot area right now is the genomic driven drug discovery, utilizing the genomic information for drug discovery, and especially in the omics data. And the second one that we are talking about, the clinical pharmacogenomic implementations. And the pharmacogenomic is the one area, hot area, that I mentioned it. Actually, there is several terms right now many famous many scientists are using sometimes using the pharmacogenomic, personalized medicines, individualized medicine, and also the precision medicines. But when we are talking about pharmacogenomic, actually the very close related with the pharmacogenetic. But the pharmacogenetic is the single term of the drugs. Response of the drugs has been caused by the single gene. But when we are talking about pharmacogenomic, is this it's a plural or many genes that has been response or influence the response of the drugs. So this, as you can see in the slide, that the pharmacogenomic actually is right now is the hot issue rather than the pharmacogenetics. And when we are talking about pharmacogenomics, I think it's one of the drug response influenced by the genetics. But the other one is like the environmental factor, lifestyles or diet is the other cause. But right now is we are we are trying to discuss about the cause of the drug response based on the genomic information that we call pharmacogenomic. Actually, when we are talking about pharmacogenomic, is one of the area that has been widely discussed right now is the genomic variations. Whilst the genomic variation is very common in our body, in every single of us, and the one we call a genomic variation actually is the synonym is with the single nucleotide polymorphisms. So this one, this is SNPs is happen in every thousands of the base pairs in our DNA sequencings. So the genomic variation is very hot issue right now. We call the polymorphism or single nucleotide polymorphisms because uh, this is based on the populations actually, because the single nucleotide polymorphism sometimes that associated with the disease, and also sometimes that also being associated with the drug responses. So this make why that medicine B might be better than medicine A. So this I think it's also an FDA right now, Food Drugs Administrations or USA right now, very clear to mention it in the every drugs that has been I think is influenced by the genomic variations. So they mentions in the many in the many websites actually. So the consequence of the genomic variations right now in the drug metabolisms, disease susceptibility, especially in the adverse drug reactions or adverse drug events, this is the other hot issue right now that caused by the genomic variations. It is an important also in our one of our ethnics in our nations in Indonesia. We call the suku Bajo. We call here the ethnic is the suku. So one of our ethnics amongst more than 300 ethnics in Indonesia, they have habits when they are find the fish. They are when they find the fish, they are trying to find without the equipment. They just use this kind of the equipment, and then they can live underwater. They can breathing underwater is more than 10 minutes without breathing, without the take the oxygen gains. So this is his name is Mr. Rohani. Mr. Rohani hasn't authorized amongst 30 years old, so very old, but they can survive underwater more than 10 minutes. It is important, and it is a very interesting study that based on the University of Copenhagen by Melissa et al. They found that two genomic variations that has been influenced in this ethnic or in this suku. And then what is the correlation? The correlation is in the increase of spleen size that making that they can live under waters more longer rather than the normal of the humans. So it is an important, I think, is, for the future of our research to elaborate and also to identify the kind of the genomic variations. So in our area in our pharmacy right now, so we call the personalized medicine or precision medicines, whilst in the other in the previously we call one dose fits all approach, when we are given the dose to the patients equal all of them. But right now we can give the dose to the patients based on the metabolism type of the patient, like the poor metabolism. If the patient has the poor metabolism, they should do decreasing the dose. In contrary, when the patient has a rapid metabolism, it should be increase the dose, because if we give with the normal dose, they will not respond to the patient. So this is the issue we call the personalized medicine as well as the precision medicines. So so William Osler did said that the variability is the law of life. If the piece of the patient, if the piece of every single of us has a difference, well, how about the molecular? Of course they will different also. It is important. So about the precision medicines, is the precision medicine actually is come up around 2015, while the presidents of Obama presidents of the United States said that tonight I'm launching a new precision medicine, since his statement that many scientists, if we see the research after Obama mentioned it about the precision medicines, all the scientists using the precision medicine rather than the personalized medicine terms. So what is the precision medicine? Actually, the precision medicine is combined the variability of genes, we call the genomic variants, and also the environmental factor as well as the lifestyle to see whether the drug has the in the influence of the drug response based on this kind of the variability in genes, environmental factor, as well as the lifestyle of the patient. So right now, the precision medicine is the very hot issue, and they using many omics data to target to target the disease, as well as to find the new candidate of drugs. So when we are trying to using the pharmacogenomic concept, we will decreasing the admissions as well as the expenses of the drugs, the cost of the drugs, and also we can enhance the safety, efficacy, and also the compliance of the patient. So this is very important. Of course, it will be very good outcome of the patient. So when we are talking about the actually there is the concept of the drug like pro drugs and also active parents or active drugs. It will be influenced with the concept of the pharmacogenomic. Like when we are talking about the active drug, the poor efficacy is the extensive metabolism. But when we are talking about the pro drugs, the poor efficacy is the poor metabolism. So it before we are going to trying or to test to the patient, of course we should to know about the type of our drug, whether the pro drug or active parent drug. So right now, when we are talking about technology, it's very developed, very fast developed in all area. When we are traveling, I think several decade ago, we can traveling by the car or ships or the trains, but now we can traveling very fast with the plan, I think. So it is same with the discover or discovery of the human genomes, discovery of the genome. But the consequence right now is the cost is drops. So it will be more possibility to implement it in the near future. Like when we are talking about the cost of the genome for human genomes at the several decade ago, it will very expensive, right? More than $100 million. But right now, it's very, I think it's a very cheap, almost 1,000. It will be more possible, I think, in the near future will be drops more a dropper rather than 1,000 US dollar. So right now, when we are talking about the, I think the genomic variants that influence the drug response, we call the pharmacogenomic, you can utilizing several website like the CP guidelines, the PUIG, PAMGKB, PAM4, and also the PGR on. So based on this kind of a website, you can utilize this kind of the website to identify what kind of the genome or what kind of the coding genes that influence the drug responses. But right now we don't have time to elaborate more details one by one. And we see here is the drug metabolism is influenced by enzyme that we call also the is sometimes the genes, and this kind of the genes was distributed in our body, and we can see here is one of the gene we call the CYP2C19 was a very influence. It was very different in every populations like the uh, like in East Asians as well as the Caucasian patient, it will be the difference profile. So this will be influence in the adjusting the dose of the patient. So we see here right now the pharmacogenomic implementations like the several drug that has been widely it has been widely used in the clinic, like the clopidogrel has been influenced by CYP2C19 warfarins, and also I think it's very common right now is the HLA about HLA-B for the abacavir in the HIV patients. We see here, for example, is very simple examples, and it was widely used in the clinic right now. When the patient has an HLB star 5701, it will be uh, possibility to the patient to got the hypersensitivity. So when the in the before the patient's given the abacavir, it is better to test the gene of the patient. If the patient has this kind of genes, rather we are not recommended to administer the abacavir. So that is very important to avoid or to uh, to avoid the patients in order not got like this kind of the conditions or also the situations. And right now I think we are still fight with COVID-19, right? And I think COVID-19 also is a very good opportunity for us, especially our background in the pharmacy, to identify the or to investigational of the treatments of COVID-19 based on the pharmacogenomic perspectives. As we can see here is that so many, I think right now that so many research area, they utilizing several of the genomes database like the PAMGKB, scientific guideline, PAM4 to identify to trying to identify whether the drug has been influenced, the response of the drug has been influenced by the genome or by the gene of the patient. So this is my uh, publication that has been utilizing the genomic informations for identify whether the patient has in the whether the genomic variant has been influenced in the populations. So the other one, I think, our study is about the evaluation of genomic factors that been influenced in the progressions of the hepatocellular to in patient with the chronic hepatitis B patients. And the other one right now, I'm focusing on the drug reproposing while we utilizing the genomic variants as well as that we integrated with the bioinformatic approach to discover new drugs, or we utilizing all drug for new indication that we call the drug reproposing. So this is the other ones that we are trying also to connect it several functional annotation in the genomic variants to identify the drug reproposing for hepatitis B, and has been published in last years, as well as that for the TB or tuberculosis, whether we collected all the genomic variants associated with the TB associated genes, and then we prioritizing the gene as the candidate drug targets, or we call drug repurposing for tuberculosis. And the other opportunity right now is we are as the pharmacists, I think, we have a very crucial role in the pharmacogenomic implementations, not only for the pharmacokinetic and pharmacodynamic area, but also for education to the other healthcare and the counseling to the patients, as well as that we should to know the drug-drug interactions of the every drugs, and also the interpreting clinical pharmacogenomic test results. So this is area is very promising for pharmacists in the future. So of course that we have constraints. When I identified in my country in Indonesia, we still have the barriers in the implemented of the pharmacogenomic because of the facility, because of this the cost is not covered by our national insurance, and also we have still many ethnies. We have around more than 300 ethnies, so it's not covered by the research area. So this is the opportunity as well as that constraints in the future. But we still have the hope and for the future to more faster to identify where this genomic variant has been influenced by in our population. So another one that we are trying to do right now, what we are trying to do as a pharmacist, of course, we can utilizing the many genomic information that has been widely used, and also we can use it freely available database that we can use for identify the drug discovery as well as the drug development. So I'm trying to know to find in the PubMed database whether the role of pharmacist and associated with pharmacogenomic. I'm trying to write it down the pharmacist and pharmacogenomics. I found around more than 300 papers. It means that they're very promising for the pharmacist to develop the pharmacogenomic. We can see here is that so many papers right now explaining the pharmacist's role in pharmacogenomic for the implementation in the in even in the research and also in the clinical area. I'm trying also to using the posterior study to see the trends of the pharmacists utilizing pharmacogenomic in the field of the genomic area. We can see here is the yellow color. It's mean that a very hot issue. We can see here pharmacogenomic testing is very close related with the pharmacist's role in near future. So of course that we can conclude it what we are discussing for today is the we are actually right now in the post-genomic era that we call the genomic medicine or we call the clinical genomic implementation in the even in the research and also in the from the bench to the bedside, and also the pharmacogenomic has a crucial role for the pharmacists. And of course, we are the for the pharmacists, we are at the forefront of the pharmacogenomic testings in near future. And also the clear implementation of pharmacogenomic, of course, that will not only feasible but also the effective, although it is still long way to implement it in the clinic. But we are again that I'm saying that the pharmacist has a crucial role in the pharmacogenomic as well as that the utilizing the omic data for many purposes, including for the research as well as that for our opportunity to implement it in the clinic. So this is my books that I am writing that I wrote with my teams about the toward precision medicine. So the last, I think I would like to say thanks to especially to Dr. Dilfreds and also the teams in the Moga Pharmacy in the India. Thank you once again, and then I think we can go to discussing session. I think, and then I send it back to the Dr. Dilfred.
Very thankful for your presentation. The presentation was indeed very nice. We have discussed about the basic concepts of genomics, pharmacogenomics, as well as what is the role of pharmacogenomics in clinical implementation of precision medicines. And what we have discussed about is the basic era of pharmacogenomics. We are here in the post-genomic area, as we discussed, and we are the forefront. Basically, the pharmacists are the forefront of pharmacogenomics. And this is one of the best statement that I have ever feel in your presentation. And regarding the presentation, sir, we have already published very excellent papers in that area, around 4 to 5 papers with international repute as well as good impact factor, and that demarcate your excellent ability in your particular topic. And you are already said that the genomics is one of the most complicated subject till now date, and the understanding of this subject and the clinical translation of this subject from the R&D to public needs, that is basically a forefront. And you have excellent explained everything. So I am hand over to the audience of that. So anyone having that question, you can discuss with sir. Any question from the audience?
We are live in YouTube as well, right?
Yes, sir. So actually, this will be recorded. That the particular topic will be recorded now, and that the recorded lecture will be uploaded to our YouTube web page, sir. The ISF College of Pharmacy web page. So soon it will be uploaded.
Okay, Dr. Pushpa.
So I'm really thankful to Muhammad Irham sir for the valuable time and the contribution, sir. Sir, you have given your precious time to the busy schedule and give you the informative talk about the pharmacogenomics as well as the precision medicine and the role in the clinical implementation. I'm really thankful to the University of Indonesia and your dean, ma'am, for providing us the permission to talk as well as you give the permission to present this lecture. I am really thankful to the university team. I am really thankful to the management of Indonesia. I am really thankful to our management team of ISFCP, Professor G.D. Gupta sir, Director and Principal of ISF College, Professor Narang sir, Vice Principal, as well as the Chairman of the college, Professor. And I'm really thankful to everyone for the joining us. Thank you very much.
Thank you, Dr.
Thank you very
Gupta.
much, sir. Thank you very much.
It's my pleasure.
तारीख है या पहली तारीख है? हाँ?